Now, one thing I want to mention here, another important point that is quite different from this, but it is important. Let me explain. Yes. As I have mentioned that, let's suppose this is a cell. This is a cell. And we know that a cell is actually surrounded by lipid bilayer because plasma membrane is lipid bilayer. Now I have mentioned that the bilayer, let's suppose the outer layer of the lipid molecule is red in color, the outer layer of the lipid molecules, and let's suppose the inner layer of the lipid molecules is in green. So this is the inner layer of the plasma membrane. Why I am demarcating these two layers into different color? It has importance. Now we call the inner layer of the plasma membrane, this green layer, okay, that face the cytosol, we call cytosolic leaflet. Cytosolic leaflet. And the outer layer of the plasma membrane is called exoplasmic leaflet. Okay. Now, why it is called cytosolic leaflet? Because it faces the cytosol, because this is the cytosol. So as this layer faces the cytosol, it's called cytosolic leaflet, and the outer layer is called exoplasmic leaflet. Names differently. Exo means outer side, so it faces the outer side of the cell, so it's called exoplasmic leaflet. Now, in my basic facts on plasma membrane, I have discussed that in case of eukaryotic cells, there are many intracellular organelles like nucleus, endoplasmic reticulum, Golgi bodies, endosomes, lysosomes, peroxisomes, mitochondria, chloroplast. So these are all intracellular organelles, and these organelles are also surrounded by membranes, and we do not call it plasma membrane, just biomembranes or membranes. Now let's suppose one example of lysosomes. Let's suppose this is a lysosome, an internal intracellular organelles. Now I have also mentioned that liposomes or any intracellular organs are also surrounded by bilayer of membranes. So there will be two layers that surrounds the any organelles. Let's suppose here I am giving one example of the liposomes. Okay. Now the layer, the outer layer and the inner layer will be different here. The inner layer in case of organelles will be red, and the outer layer of the organelles will be green in color. Okay, completely opposite to the plasma membrane. This is the color coding of plasma membrane, but this is color coding of for the intracellular organelles. Why? Because I have mentioned that this green color layers actually face the cytosol. So here also the green color layer will face the cytosols. Okay. Why? Because the composition of the lipid molecules in the cytosolic layer, cytosolic leaflet, will have to be remain same for the plasma membrane and also for the membranes of other organelles. It has to be understood. So the color coding here will be that the outer layer will be the cytosolic leaflet. So the chemically this is the cytosolic leaflet, and this equivalence is called topological equivalence. This equivalence is called topological equivalence. Okay, and this red layer is actually equivalent, topologically equivalent with the outer exoplasmic leaflet. Now, how do we know, or how could we actually explain this fact? It can be easily explained with the help of one process that performed by cell is called endocytosis. So I can explain this with the help of endocytosis. What happens when endocytosis occurs? Basically, invagination occurs. That means if any molecule is present outside, let's suppose this is a molecule present outside and it wants to enter inside, then one process called endocytosis occurs, by which cells actually engulf the molecules. When it engulfs, what will happen basically, it forms a invaginations. It looks like this. It looks like this, okay? And see, the red layer of the membrane now will be here, okay? This is simple, okay? This is the continuity. Now let's suppose this molecule is present here. Now this molecule has entered, so it is the beginning of the process of endocytosis. And when this pinching off occurs, that means two bilayers actually come very close and closes it on off, then it will look like this. Ultimately it will become like this. Basically, you see, it will look like this, because now the green will remain the outside, the green will remain the outside, the red will remain the inside, and inside the molecule there will be the, inside the red layer there will be the molecule, and this is called endosome. See there. So basically the inner layer of the membrane, lipid present in the membrane of organelles are very similar or equivalent. Basically they are chemically equivalent or topologically equivalent with the exoplasmic leaflet. And we know that this organelles actually, this is the lumen of the organelle, lumen, and is very similar with the outside environment of the cells, because always the cytosol face the cytosols. So this is called topological equivalence. Hope this video will help you. Thank you for watching, and always keep learning.